Let's do some questions of molecular basis of inheritance. The actual 3D structure of tRNA looks like an inverted L shape. The specific tRNA used at the start of translation is called initiator RNA. Which statement about tRNA is incorrect? There are tRNA for all 64 codon including stop codon. तो ये चीज़ गलत है. Basically यही आंसर होगा. tRNA carrying serine has the anticodon UCA. The enzyme that charges tRNA with its correct amino acid is aminoacetyl tRNA synthetase. The ribosome consists of structured RNAs and about how many protein? 80. In its active state, the ribosome exists as two subunits, a large and a small subunit. Translation begins when the small subunit encounters an mRNA. In bacteria, the rRNA that catalyzes peptide bond formation is 23S-rRNA. The enzyme like ribosome that catalyzes peptide bond formation is called a ribozyme. Translation is defined as polymerization of amino acid to form a polypeptide. The order and sequence of amino acid in a polypeptide are defined by the sequence of bases in the mRNA. The bond formed between adjacent amino acid during translation is peptide bond. The formation of peptide bond requires energy. Amino acid are activated using ATP. Activation of amino acid using ATP and linking to cognate tRNA is called charging of tRNA and amino acylation of tRNA. A translation unit in mRNA is the sequence flanked by the start codon and the stop codon. Additional sequences in mRNA that are not translated are called untranslated region UTR. UTR are present at both 5' end before start codon and 3' end. UTR are required for efficient translation process. The start codon is recognized only by the initiated tRNA. After initiation, the ribosome proceeds to the elongation phase of protein synthesis. During elongation, tRNA anticodon forms complementary base pair with mRNA codon. During elongation, the ribosome moves along mRNA in which direction? 5' to 3'. During elongation, amino acids are added one by one. At the end of translation, which factor binds to stop codon? Release factor. When release factor binds to stop codon, translation is terminated and polypeptide is released. Which molecule link amino acid language to nucleotide codon language, that is tRNA? In eukaryotes, gene expression can be regulated at the transcription level, which involves formation of the primary transcript, which is not a level of gene expression in eukaryotes, replication level, formation of new DNA. Regulation at processing level in eukaryotes refers to regulation of splicing. Lactose is termed as inducer because it regulates switching on and off of the lac operon. In the absence of a preferred carbon source, if lactose is provided, it enters cells through action of permease. A very low level of lac operon expression must be present all the time so that lactose can enter the cell. Permease must be available. The repressor of lac operon is synthesized all the time constitutively from the I gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing. In the presence of an inducer, the repressor is inactivated by interaction with the inducer. When the repressor is inactivated by the inducer, RNA polymerase gets access to the promoter and transcription proceeds. Regulation of lac operon can also be visualized as regulation of enzyme synthesis by its substrate, which is lactose. Regulation of lac operon by the repressor is referred to as negative regulation. Which of the following cannot act as an inducer for the lac operon, that is glucose or galactose? In the absence or inducer, which of the following correctly describe the lac operon state? A repressor binds to operator and prevent transcription of the operon. In the presence of inducer, which is lactose, which correctly describes the lac operon state? Inducer binds to repressor, inactivating it. RNA polymerase transcribes ZYA. The lac operon consists of one regulatory gene and how many structural genes? 3. ZYA. The I gene is derived from the word inhibitor. The I gene codes for repressor of the lag operon. The Z gene codes for beta galactosidase beta gal. The Y gene codes for permease. The A gene encodes trans acetylase. All three gene products ZYA are required for metabolism of lactose. Lacoperon is under positive regulation as well, but it is beyond the scope of discussion at this level. So yeah, that is it for this video. It was not any explanation videos, it was just question video.